nowadays in GER-MING or the things we create by ourselves, we create a lot of data, and we download sequences from GER-MING quite a lot. But many people they talk about the problems about the huge bias exists in the data bank of the from NCBI, for example. And here is a paper published a couple of years ago talking about the data availability inside the gene bank. They use the genes, the different orthologs from here to here, and they list the species name, the taxa on the y-axis. And if there's one single data point, then we'll have a little dot here. For example, there's a species, certain species has this certain gene. And of course, this is almost 10 years ago, but right now the situation is very similar actually. And it's only concerned about the green plant species, and with 13,000 sequences from the gene bank. And you will see the dot does not distribute evenly among this huge space. And they almost always concentrate on a certain point. And over here are the model systems. So on most of the genes available only for those modern model systems like Arabidopsis, rice, maize, soybeans, and so on. And some of the genes are very popular for the large-scale phylogenetic analysis. So you will see they are concentrating on the upper part and on the right. So it means that even if you want to combine the data, you will have some problem. And if you want to do some studies, you are very likely to face a lot of missing data in the middle. And how are you going to combine it? There will be a huge challenge. And even we are not worried about this data partition problem, we still have a huge problem on the missing data. So that's about the time the supermatrix and supertree approach has been developed just early in the 10 years ago. And here are the data availability situation and how we usually deal with it. And here are the species list and the taxa. And for some genes like gene1, we have a lot of sequences. This very commonly used marker, like for example, it will be ribosomal DNA or mitochondria, COX-1, for example. So we will have a complete setting for this one. But in some analysis, some people only study, for example, a small genus and using several different genes. And you can combine those data a little bit easier. But in some case, for example, some people work on a huge, if we are consider working on a huge family and of the plants or animals, and some people they only they only study some genera and using a certain gene. But some people from Japan they study some overlap taxa, but using another marker. And some people from America they also use another one. And how are we going to combine the data like the one on the right? And there are several different approaches, of course. And combining those data matches, even though they have the same taxa, and you will likely will combine the different phylogenetic signals. And so in some case, it's the evolutionary history is very obvious. It's different. Like the non-photosynthetic plants, their photosynthetic genes will have different evolutionary history than the ones can do the photosynthesis. So some people, they use this super tree approach. And the idea is that, for example, there are 3 different studies, independent studies. They all working on small part of the big phylogeny. And they have 3 different trees. And the easiest one is just to combine it, and you will have a big tree, and with a small part as representing by the specific data sets, and this yellow part representing another one. And this is the one of the approach, but in many cases it's not really easy to combine. And the new approach is using a so-called super matrix. The new super tree approach is to combine the 3 different topologies and combine into a new data matrix with some of the missing data and stating the relationship, and they will give you a new super trees. So this super tree is a little bit different from the ordinary thinking to just graph and attach to the bigger picture, but with some of the overlapping signals inside. So what we are going to do, we're to deal with tree. And for example, we have a source tree, the 2 different source tree, and one with A, D, E, F, the other one is B, C, D, E. There are 2 taxa are overlapping on those 2 trees. The D and the E are both present over there. So we want to use those things as a backbone and try to combine those 2, the information from the 2 trees. There are 2 ways to combine it, and for these are just 2 of the examples you can get. And this is a super tree you can get. It's A, B, C, D, E, F, and with this relationship. And another super tree you can construct is B, C together, and then D and A. And these 2 trees are totally compatible with the middle source tree. So for example, if you want, we just focus on the super tree A first. And when we examine, okay, so how about the relationship between A, D, and E, F? A is here, D is here, so it's totally compatible. And this is also compatible with the one on the right. You just eliminate A, it's the same. And it's the same thing for the super tree B here as well. We just cut out the B and C, will be the same as here, and just cut out A, will be the same as there. So it means that there are more than one solutions to get the super trees eventually. So if we can explore all the possibilities, and we can make another consensus tree according to it, and to see there's competing data. So alternatively, we can use the super matrix approach and to combine the data again. And this is the same example we just got. So for the sequence 1, we have A, B, C, D, E. But for the sequence 2, we have D, E, F, G, H. So there are 2 things right here. And for the others, we just put the missing data. And we use this new data matrix to reconstruct the phylogenies. We can still use maximum parsimony or some other methods, and to still making the trees according to all the data available right here. So that's one approach. But the other approach, we can just deal with the tree itself, and other than go to the super matrix. And we can first, well, basically the approach is we have to identify the common taxa like we did already. For example, we want to combine the A and B trees, so we have to identify the common taxa. It's A, C, and E. So we'll use the A, C, and E as a backbone for the further analysis. Then we add up this small part of the B1 and B2, and to see if it's here or somewhere else. And if there's no additional data, we just ignore it. And we just worry each of the the branch one at a time, and eventually we can get the tree something like this one. And the DF and MN does not overlap along the branch, so we have no idea about the forward relationship. So we just combine the forward into a new node. Okay, that's the basic thinking. And there's some other new approach called the MRP, the matrix representation with parsimony. And this is a little bit different from the previous one. This is how they actually do the tree, the super tree, but not using the super matrix. So they basically create another data matrix, but only concerned about the tree topology. And there's some approach right here. Okay, so basically what we do is once we make a tree, we just don't worry about the matrix anymore, and we have to identify the new node or the new branch or each of the node of the different trees right here, like the one on the left and on the right. So we create a new data matrix. This data matrix has a new component to let us to visualize the relationship among the different groups. For example, they will have a new components right here, A, which contains the member of 1, 2, and 3. 1, 2, and 3. For B branch, it has number 1 and number 2, but with missing data for the others. For example, for the number 6 and 7 only happen on the trees on the right. So we create a new matrix, and we can use this matrix to reconstruct the super trees accordingly and make a phylogeny out of this matrix. So we can also add up some of the weighting. Some people they use different weighting for the different branches, but that's another story. So this is the algorithm they use, but you don't really need to worry about it. And basically, they just have a different coding for different branches, and each of the node can have a collection of the taxon number, and they use this information to construct the new tree. And there's another algorithm. It's called a build algorithm. It's basically they just separate one taxon and then repeatedly to evaluate all the cluster and to resolve the relationship. So you don't really need to worry about that part. Just need to know, okay, there are two different algorithms can give you the result, and those algorithms are a little bit different in their outcome. For example, right here, and the MRP tree will have a little bit streak in their resolution, and the build tree will have a little bit better resolution in this case. So using the different algorithm, you can get a little bit different result, but the overall picture is the same. And there are several supertree methods and programs you can use, and these are just for your information. If you are really interested in that part, you can work on that. And there's some examples for, there are quite a lot. This is an old review, and there are even more taxa using examples using this super tree approach to combine it. And there are several new approaches having developed for super tree construction, and something is called divide and conquer. And basically, the basic idea is that if you are combining a lot of data matrices, then it'll be a little bit too complicated for the computer to analyze the global picture. So some approach are basically to divide it, the super matrix or super trees into a small compartments, and analyze that first, and then combine the data afterwards. So this divide and conquer. So to separate to a smaller problem and then combine it. And there are several different approaches and different algorithms have been developed. And something like the discovery methods to have a very huge data matrix and just to create a very rough phylogeny first, and then only work on a certain part of the branch one at a time, and then combine those data together. And because it basically will save a lot of computation time, because the more taxa we add, the more difficult we will be able to get the result. So anyway, this is one way to do it, and the other way is to identify a certain group of the taxa from the data matrix itself and create a small subset of the data matrix and do a conventional phylogenetic analysis. The basic thinking is the same, is to divide the problem into smaller compartments. So of course there are some criticisms on the supertree approach, because once you get into a supertree approach, it's almost only worry about the tree itself, but not worry, just bypass the matrix. And the data set is also non-independent because it overlaps quite a lot. And the MRT approach will favor larger source trees. So if a certain taxa, the certain data matrix, a certain part of it has a more complete information for those taxa, then the method will basically highly influenced by the method because it contains more information. And also the method we just introduced do not put like the if we already have the bootstrap support or jackknife support, it cannot be incorporated into the consideration. So there's another weakness. And there are some programs available if you are interested. And there are two programs, both called SuperTree. One developed by Rod Page, another one by JER-MING. And you can check on the website if you are interested. And those kind of big tree problem is problematic not only to calculate it, but it actually is to visualize as well. Once you get to over 10,000 or 1,000 data taxa, then you will have very difficult way to actually see all the little nodes. And Michael Sanderson is the middle one right here. Is the one who always doing this kind of things, and we have a lot of computer monitors putting together to actually see the small part of the trees. So this is a kind of an interesting way to see it.